Welcome, everyone, to today's congressional briefing, Accelerating Breakthrough in Brain Research Through NIH Brain Initiative. I'm Jen French. I'm the board chair of the American Brain Coalition and the co-founder and executive director of Neurotech Network. Today's briefing is hosted by Senators Eric Schmidt and Richard Blumenthal, as well as the Congressional Neuroscience Caucus co-chairs, Representatives Morgan Luttrell and Mike Thompson. Senators Schmidt and Blumenthal have been strong champions of the BRAIN Initiative. Most recently, they've been leading the Senate Dear Colleague letter, calling for robust and sustained investment in this important program. Representatives Luttrell and Thompson have also been steadfast champions, hosting briefings over the years and leading the House Dear Colleague letter in support of neuroscience research. In conversations with the Senate staff, it became very clear to us that this is the right moment and the right venue to underscore the value of sustained bipartisan support for the BRAIN Initiative. We also know that it's not always easy to come over to the House side, so we just thought it would be especially important for us to be hosting this briefing today in the Senate. This briefing could not be possible without the American Brain Coalition partners that are sponsoring this event today. That includes the American College of Neuropsychopharmacology, the Dana Foundation, IEEE Brain, the Kavli Foundation, the Simons Foundation, the Society for Neuroscience, and the Society for Biological Psychiatry. And on that note, I wanted to say, 28 years ago, I had a spinal cord injury that left me paralyzed. A year and a half after that, 27 years ago, I was implanted with an experimental neurostimulation device that allows me to stand and use my paralyzed limbs. I'm still using that device today, and that investment in neurotechnology has drastically changed my life. I think what I've learned over that period of time is that the innovation ecosystem does not just consist of scientists and researchers. It consists of industry representatives, it consists of clinicians, of hospital systems, of payers, of investors, of funding agencies, and patient advocates. And all of them help to make this ecosystem possible. It's those living with neurological conditions, their family members, their care partners, their loved ones that are really important in the role of scientific discovery and innovation. And with that, I would like to introduce you to our opening remark from our dear friend, Tish Havel, who is the founder and CEO of the Brain Donor Project. Tish.
Thanks, Jen, and thank you all for including me in this auspicious group. I should tell you to start, I am not a distinguished government representative, nor an accomplished scientist. All I did was build a soapbox to stand on from which to preach the gospel of brain donation, and I did it at the behest of brilliant scientists. So it was shortly after, I've got my story too, many of us do, it was shortly after my dad was diagnosed with Lewy body dementia. And we happened upon the concept of brain donation while we were trying to learn about his disorder. When we eventually donated his brain for neuroscience research, people in our circle wanted to know more. So we dug in a little bit, learned a little more, and started this nonprofit, the Brain Donor Project, to raise awareness of what these scientists...
Hello? Better? I'm sorry. Hi, everyone. My name's Tish. Anyway, what these scientists kept telling me, that donated human brain tissue is the most precious resource in neuroscience. It holds the answers. It just does. And because of projects like the Brain Initiative, the tools are being developed to get to those answers. We need them desperately. The WHO estimates that 1 in 3 of us either has a neurologic disorder now or will develop one in our lifetimes. So here's how that's going to shake out. If in your circle, from the grandparents to the grandchildren to the siblings, cousins, all the in-laws, no one's been diagnosed with a neurologic disorder, you are incredibly lucky. And there's a very good chance that luck will run out. It could be a dementia. That is the most often reported diagnosis that we hear from families working to make arrangements to donate the brain of a loved one. And that's going to get worse because new dementia diagnoses are projected to double within the next couple of decades to 1 million per year, fueled by an aging population. Or it could be somebody with a serious mental health issue. I talked with attendees about this last month at the annual meeting of the National Alliance for the Mentally Ill. It's pretty widely known there that donated brain tissue will lead neuroscientists to the source of those disorders, a very special category of biomarkers that will finally move much of this field beyond hit or miss efforts at treating symptoms. It could be Parkinson's too. That's another one that's very widely reported. The focus is a tiny little piece of the anatomy in the midbrain that misbehaves. It's called the substantia nigra, so tiny, 25 of them could fit in a golf ball. So we could use them up, right? Depletion of those tissues stops critical research. Then on the opposite side of the diagnosis game of chance are the rare and the ultra-rare, often neurodevelopment disorders. The urgency of arresting a brain disease that is doing irreversible damage to a child cannot be overstated. It is these warrior parents who make the gut-wrenching decision for brain donation so that other families have a chance of winning this horrible race against time. Each of these carries its own agony for the individual and his or her family. And in the 10 years we've been operating the Brain Donor Project, we've been telling their stories to help people understand the critical need for this precious tissue, so far helping about 30,000 people either donate a brain or make arrangements to do so when the time comes. And we're working to dispel a lot of myths about that too. Like many of you may have thought, it is not part of organ donation. Checking the box on your driver's license has nothing to do with brain donation. You have to make separate arrangements. And no, it's not the same as anatomical body donation. Your brain doesn't get used for neuroscience research that way, but there is a way to do both. No, it isn't disfiguring. And very importantly, no, you most certainly do not have to have a diagnosis. So-called unaffected controls or healthy brains are needed in every single study. So here's an uplifting little perspective. We're all going to die. What we don't know is how or when, but it's going to happen. We can plan for certain things. Many of us give a lot of thought to our stuff and what will happen to it when we're gone, and our money if we have any left. Yet our greatest legacy-building gift, the source of our humanity, some people say, arguably the most valuable resource in science, since there's no substitute for it and we can't just make more, well, it should probably get a little more consideration. So I preach this gospel of brain donation on behalf of the people who do the hard work of figuring out why the circuits misfire, why the tangles get a little too tangly, or how to repair gene anomalies that shorten or make impossibly difficult the lives of way too many people. Look, every neuroscience breakthrough begins with a question, and the answers come with scientific exploration. We are already on the brink of revolutionary discoveries. Brain donation, about which I am a little passionate, is part of that equation, but it does nothing without sufficient resources to explore it. Jennifer, I'll turn it back to you.
Thanks,
you, everyone.
Tish. Thanks, Tish. You offer us a really important partnership and collaboration, so we appreciate it. And now it's my pleasure to turn it over to Dr. John Nye, who is the director of the NIH Brain Initiative. to lead today's discussion. John.
Thank you so much. Thank you all for being here. Thank you to Senators Schmidt and Blumenthal for co-hosting this briefing. As Tish said, none of us will really escape the burdens of neurological or neuropsychiatric diseases. 1 in 3 humans on the planet at some point in their life will live with a disorder of the nervous system. That translates to, in our country alone, over 100 million people. At a huge personal and societal cost, but also at a substantial economic burden. There's a lost economic activity of about $1.5 trillion a year in our country alone. Now, the human brain is really just this marvelous organ. It's the most complex in our body, 86 billion neurons or nerve cells that carry information to each other through trillions of connections. There are also other cells in the brain that modulate function. We're going to hear about some of that from our esteemed panelists. And together they give rise to everything that makes us human: our emotions, our thoughts, our ability to love and receive love, to be joyful and sometimes to be less so. But its very complexity is what makes it vulnerable to disease. Stroke, childhood epilepsy, degenerative diseases like ALS, Parkinson's, Alzheimer's. And these degenerative diseases are also on the rise because the good news is we're living longer, but one of the major risk factors for some of these diseases is age. So it's a big problem we really need to solve. And of course, there are also devastating neurodevelopmental disorders like autism. So I've been at the NIH for just over 6 years. It was the fall of 2019. I just decided to take on this job. I was a university professor, arguably one of the best things one can do in our field. And very close family friends of ours, their son was getting married. Now, many of you are of a certain age. You might be familiar, or some of you with young families. You become very close with families. Their kids grow up. They go to different schools. You might stay in touch with some, but you might not see each other for a couple of years. And this was a great opportunity for us all to get back together again. So I was sitting next to a very good and old friend, Deborah, who I hadn't seen for a couple of years. We kind of lost touch. Our kids went to school together. And she asked me, well, what are you up to? And I told her, I said, look, I'm giving this great thing up at the university. I'm going to go work at the NIH to do this thing called the Brain Initiative. She said, what's that? And I said, well, we have all these great tools to understand how the human brain works, what goes wrong in disease, and maybe we can find a way to fix it. Deb is an artist. She's very, very sharp. Would not let me off the hook. And she said, well, you got to tell me more. Be more specific. So I tried a little bit more. She said, no. Be more specific. I have a vested interest. We tend to talk with our hands, and I noticed at that point she had her hands in her lap. She was suppressing a tremor. She had Parkinson's. So for her, for all of us, for our family members, 1 in 3 Americans, everybody has a family member, a loved one, where the cure cannot come fast enough. And that was 6 years ago. We're making progress. I thought, honestly, that we could find cures for some of these diseases in our children's lifetime. I can honestly say right now that I think we might get it even closer, maybe in our lifetimes. We're making breathtaking progress, in part because of some of the great research that's being done, represented by our panelists here. And it's really due to their creativity, their ingenuity, their passion, their absolute passion to get all this done. And this is what the Brain Initiative is all about, giving us the tools and resources to understand how this incredibly complex organ in our body works and to understand what goes wrong in disease. We have a saying around the Brain Initiative, you can't fix it if you don't understand it. And our mission is to understand it and to find new cures and therapies for these devastating diseases. So as I said, we have this amazing panel of scientists spanning the spectrum from basic discovery. You have to understand how this thing works. You have to translate it, so you have to know how to fix it. And then you need to make it happen in the clinic for the benefit of society. So we have with us here, let's see, on this end, Dr. Patricia Janik, who is the Bloomberg Distinguished Professor in the Departments of Psychological and Brain Sciences, and also part of the Kavli Neuroscience Discovery Institute at the Johns Hopkins School of Medicine. And she's also the incoming president, the president-elect of the Society for Neuroscience. This is an international society with about 30, how many thousand members? A lot of members.
Something like that.
So she's, you can tell, very prominent in the field for good reason. Yep. To my left is Dr. Ed Lean. He is an Executive Vice President and Director at the Allen Institute in Seattle, and we're going to hear about it. He's just launched this very exciting initiative called the Brain Health Accelerator. And then to my far right is Dr. Sandeep Robert Datta, who's a professor in the Department of Neurobiology at Harvard Medical School, and Bob is doing some amazing work that we're going to hear about in terms of getting quantitative information about animal and human behaviors. And next to Dr. Datta is Dr. Martha Morell, who's the Chief Medical Officer at a company called NeuroPace. And this, again, is very important for us to get these discoveries into the clinic, and part of getting that done is forging the right public partnerships to get these devices into the commercial arena. And she's also a clinical professor of neurology at Stanford University. And then, last but not least, is Dr. Marina Picciotto, who's the Charles B.G. Murphy Professor of Psychiatry and Professor in the Child Student Center of Neuroscience and Pharmacology. That's a lot of departments. That's too many. And not done, also a member of the Kavli Institute for Neuroscience at the Yale University School of Medicine. Dr. Picciotto is also past president of Society for Neuroscience and the current president of the American Association for the Advancement of Science. So, as I said, we have a great opportunity to learn about how to get us closer to cures by starting with basic discovery, thinking about how to translate this into the human arena, but then also getting it into the clinic. So Dr. Picciotto, tell us a little bit about your work in developing how it contributes to life-saving treatments for tobacco addiction.
Absolutely. Well, I think none of us would imagine that lung cancer is a brain disease, but in fact, a large proportion of lung cancer patients are smokers who tried to quit and could not, or who got hooked very early and then kept smoking, my own father. And so I'm a very basic scientist. I work on an acetylcholine receptor, but it happens to be called the nicotinic acetylcholine receptor because it is the primary receptor for nicotine in your brain and body. And I'm really excited to talk about it because this is one example where very basic science got translated into the clinic and has already, in the last probably 15 years, saved hundreds of thousands of lives. We started working on these receptors because they were interesting molecules in how our brains and bodies work. And then it was discovered that they were the targets for nicotine, and very basic research in cells, and then in live animals, mice, and in rats, led to understanding of the pharmacology of these molecules, and led to a very specific drug development of targets to tweak these particular molecules just enough to compete with nicotine, but not to put people into withdrawal. And that molecule was then commercialized by Big Pharma. Great. They have medications now that have helped people go from about 6% quit rate for unaided smoking cessation attempts to, in combination with other therapies also developed in the same way, up to 30% success rate. Now, that may not seem like a lot because you'd like 100%, but for something that is as lethal as smoking, it has resulted in, from a conservative estimate, maybe almost 300 or 400 million lives saved. And that came purely from a number of projects across the disciplines of neuroscience trying to understand the basic function and the cells and the organization of the brain and the behaviors that lead to compulsive nicotine seeking.
Thanks. So I never thought of lung cancer
As a brain
disease. But, you know, there was also a change in the 60s and 70s looking at behavioral disorders as a brain disease. And this is very, very, very important for us to think about: depression, OCD, PTSD, and the like. Dr. Janik, tell us a bit about your work in the area of alcohol use and abuse and how this ties in with having to understand neurobiology.
Yep, happy to. First, I just want to say another quick thank you for giving us the opportunity to tell you about how Brain Initiative and NIH funding is leading us to new discoveries and new treatments for people. We're really, really honored to be here. So I'm an addiction neuroscientist like Dr. Picciotto. And addiction is really tough. And so I want to talk about not this outstanding success story, but some things that still remain for us to work on, and some things that the Brain Initiative tools help us to work on. So addiction is tough because no matter how hard people try to stay sober, they often relapse. So they suffer from an uncontrollable urge to take another drink of alcohol or to take another dose of drug. And this uncontrollable urge, this is a feeling that's produced by the brain. So we know this. So scientists have taken humans and put them in brain imaging machines during the times when they're strongly craving to take a drink or to get another hit of drug. And we've seen the brain circuits that are activated by this. So really excitingly, we know the brain activity that's responsible for this feeling, this urge to take the drug that can lead to relapse. But we don't know how that works. How does that start? How is that triggered? And so what we hope to discover by studying this are the triggers for these brain circuit activations that lead to relapse and how that's translated into a person actually making the decision and going to get the drug. And in some cases, the very hopeful cases, how does a person suppress the brain activity that is the urge to take a drug and stay abstinent? So we can study these processes to try to lead to treatments by looking at the biology of it. How does it work in the brain? And the tools of the Brain Initiative are helping us do that in animal models. And so I'd be happy to talk more about the details of the tools and how we use them, but I just want to give you a picture of this problem and how that's something in the future that we're tackling with the new tools from the Brain Initiative.
That'll be great. We'll certainly have time to get our science geek on. Don't worry about that. You know, the research that you both have been doing is amazing. And as we think about the biology of the disease and treatments, of course, how do you know you're actually helping somebody, right? You go to the doctor, they take your blood pressure. That's what we call a biomarker. EKGs, we've seen huge advances in cancer therapies because cancer researchers have given us what we call biomarkers. You can tell what kind of cancer you have. You can tell if it's responding to treatment, and you can pivot the treatment as things occur. But one of the frustrations in neurology and neuropsychiatry is that, how's the person feeling? Many of you might have gone to the doctor with a sore something or other, and you might have been asked, what's your pain on a scale of 1 to 10? That's pretty subjective, yeah? How well is a person responding to antidepressants or other drugs to control their mood? So Dr. Datta has come up with some really ingenious ways of measuring animal behavior and also human behavior. So could you tell us a bit about that and how it's going to help us to not only develop drugs, but actually to treat people?
Yeah, thanks, John. First of all, let me add my voice in thanks. I'm really grateful to have the opportunity to do science, and the Brain Initiative has been a really important part of that and in helping our science move forward. So thanks to all of you for that. I think it's useful to start by just thinking for a moment about what our brain does. Like, we think as humans that our brain lets us think and remember and have emotions and feelings. But the first brains interacted to do a very simple thing. They evolved to do a very simple thing, which is to let animals behave, to let them act in the world, to let them make decisions and make movements that reflect their needs and desires, and to allow them to survive. And so a lot of what my lab does and thinks about is, how does the brain take decisions and turn them into behaviors? And to explain why that's so important, I'd like to tell just a quick story about my dad. My dad is in his 90s, and he's living with Parkinson's disease. And if you ask him when he thought he became symptomatic of Parkinson's disease, he says it was about 5 years ago. If you ask my mom, though, she says she noticed changes in the ways that he was moving his body about 10 years ago. My dad's doctor is brilliant. It's this guy named Carlos Portero Callao, and he turns out to be a Brain Initiative-funded physician scientist, and he sees my dad all the time. And if you ask Carlos when he noticed my dad beginning to change, he said it was about 15 years ago. And if you ask him, what were you noticing? Carlos will say that it wasn't anything that was in the textbooks, but just in the way that he moved his body, in the subtle ways that he answered questions, he knew something was going on. And so my lab's research program really is to build artificial intelligence tools, to look at animals and to look at people, and to ask how their movements might be changing as a consequence of disease, and to use those as biomarkers, to use those as early clues for when disease onset is happening, so that we can both know when something has gone awry and we can track its progress over time. These sorts of tools are super important in developing drugs preclinically. We can look at animal movement and test drugs and ask how those movements have gotten better. And they're super important for understanding how different individuals might have different varieties of disease. These kinds of biomarkers are really important, I think, in the future for personalized medicine. And I'm happy to talk more about the methods later.
Yeah, that's great. Thanks. So Dr. Morell is our practicing physician here. Dr. Morell, tell us a bit about your experience with childhood epilepsies. What is it? What is the burden on society? What is the burden on families and the frustrations and challenges of treating these devastating conditions?
Yes, I am a neurologist who has been taking care of people with epilepsy my entire career, adults and children. Epilepsy affects 1 to 2 out of 100 people. It's common neurological disorders. And 30% of people with epilepsy do not respond to medications. So these were the patients I have been treating or was treating my entire life, and doing research to try to reduce the burden, but recognizing that in many patients we just didn't have any options. So I heard about a small group of engineers who came from cardiac, and they were interested in using the principles of cardiac defibrillators or cardiac pacemakers to treat this electrical disorder in the brain. I had an endowed chair at Columbia. I was very happy. But I thought if I can become part of this, then I could perhaps contribute to something that would help the patients that I can't help. And this is a very personal disease. Many of you know somebody who has epilepsy. It impacts the community around the patient, families, and friends. It limits educational and occupational opportunities and is associated very oftentimes with injury and a much higher risk for death, sudden unexpected death in epilepsy. So there is a sense of urgency. I felt a sense of urgency that I do something that mattered for these patients in my lifetime.
Thanks. And we're going to hear a lot about what that is. It's really, truly remarkable. So, Dr. Lean, you've embarked on an amazing journey creating an atlas of the human brain. Again, 86 billion neurons, and developed many of the tools to understand the parts of the brain and how they function. And I view that information as our Rosetta Stone. So tell us the importance. Why do we need to create a map of the human brain, and what's the future going to look like as a result of getting this information?
Yeah, thanks, John. And I'm really pleased to have the opportunity to speak here today and to convey, I think, the importance of really basic, fundamental reductionist research. In the human brain, we have a complexity problem and an accessibility problem. As Tish mentioned at the beginning, this can only be done by looking at donated brain tissues, but it's also by far the most complex organ in the whole body. We can think of this as a cellular organ and also as a circuit. The cells give rise to the functional circuitry that allows us to do everything we do as human beings. And we have a really poor understanding of this in humans. Most of this work is done in model organisms, which bear very strong resemblance, but not at the level of the very fine details. And I think really the Brain Initiative has had an enormous effect on the field through its strategy of first stimulating new technology development. Everything we do is really limited by the technologies we have at hand. And then putting them to use at scale to really create transformative references. And what I'd like to tell you about today is our work through the Brain Initiative Cell Atlas Network, or BICAN, where we've really tried to take technologies that let us understand the cellular makeup of the brain and get a complete map of all the types of cells that make up the brain. I mentioned technologies. In this particular case, the technologies that have turned out to be transformative came from another field. They came from the field of genomics and a derivative of the Human Genome Project. Whereas in the past, we had defined cells by their shapes or their firing patterns or their connectivity, now we can define them by their genes, the genes that they actively use that give rise to their properties. And if you can apply these techniques at scale, as we've been able to do at the Allen Institute as part of BICAN, you can completely map the entire brain and get a description of the entire system. This is like having the Human Genome Project available to us and Google Earth all at once. And this is a transformative resource. All of a sudden, we understand the system at this level, and we can start to understand then what happens in disease at the level of cells and circuits. And so I'll get into, I think, a bit later about how we can use that map immediately now to study disease and get at the types of cells that are most vulnerable in disease, and also that we can manipulate now. And the other side of this that's come from this work is that we can now have the tools to genetically target any kind of cell in the brain. And so if we can identify the cells, we can target the cells, and that opens up a whole new avenue for genetic therapies for various kinds of brain disorders. I think this is so potentially transformative that I've bet the rest of my career on this. We've just started a new initiative centered at the Allen Institute called the Brain Health Accelerator that really takes this whole suite of tools developed by the Brain Initiative and is turning them into now a huge discovery effort to try to understand vulnerable cell populations and push it all the way to cell and circuit level therapeutics.
Yeah, I hadn't thought of it that way, that you've bet your entire rest of your career, but you have. This project that you've referred to, we launched in 2022 or so. And I was kind of betting, it's been said the only people that need to worry about their reputations are chefs, but I bet my reputation, the reputation of NIH on this project, and I gotta say, it's been truly breathtaking to see all that progress. So let's dive in a little bit. So could our two addiction experts tell us, let's get some details about how we got from A to Z in terms of the initial discovery to helping humans. Which one do you want to start?
Go for it, sure.
So this is a story that's interesting in that it started with the torpedo electric fish. And the torpedo electric fish has these wings with batteries in them, basically. And if you touch those batteries, you get a 40-volt shock. It's how they stun their prey. And the way they do that is by loading up those batteries with nicotinic acetylcholine receptors. It turns out that was a great source for biochemists who wanted to really get into the nitty-gritty details on the structure and the function of this crazy protein. We have it at our muscles, in our muscles. It's how nerves and muscles communicate, absolutely essential. And all of those details that came from the torpedo electric fish were then used to ask about the structure of our receptors in our muscles. And that information was then used to figure out the structure and the function of another family member of the nicotinic acetylcholine receptors in the brain. Without the torpedo electric fish, we couldn't have done it. Without our neuromuscular junction, we wouldn't have been able to imagine what was going on in our brains. And with the advent of cell type selective technologies, we wouldn't have been able to figure out what those structures do to the function of our brains. And that whole journey was absolutely essential to understanding why we take this tobacco leaf, roll it up, set it on fire, and put it in our mouths. A lot of people now vape. They've switched from smoking to vaping. Problem there is it's still nicotine addiction, and we won't know for 30 or 40 years what that's doing to our lungs, our tissues, or our brains. And in fact, the whole array of things that these receptors do in our brains, because of tools that allow us to know what cells, what circuits use these particular molecules, we know that it's important probably for early life attention, so relevant to attention deficit hyperactivity disorder. We know what it does to circuits involved in mood. So the relationship between smoking and depression has really developed from understanding this is a very basic molecule that came from the torpedo electric fish. So that journey, which took many, many different labs and had to depend on funding before it was clear that even there were nicotine receptors in the brain, depended on the faith of the public, of you, that science of that very basic kind might lead to an outcome that could be helpful to humans.
Yeah, you know, one thing I've learned over the years is that you never know when the next big hit's going to come from. But if we give scientists the tools to pursue their passion and creativity, we come up with these big wins. The big wins are not predictable. We just have to provide the ways of doing it. Also, as you mentioned it, when I was a grad student, there was a lab studying torpedo. And as kind of a mischievous young guy, I never went near those tanks. I was scared to death of them anyway. So, alcohol addiction. How has the studies of model systems and the like, how has that really advanced our knowledge?
Well, one thing that the audience might not know is that animals take drugs that humans take. Animals, besides humans, will drink alcohol and get drunk. They'll take any other drug that humans abuse. So what that means is there's enough similarity in the brains across mammals up to humans and in how we respond and how we like alcohol and drugs that we can use these animal model systems to study and understand the neurobiology of alcohol action in the brain. So in order to really know where alcohol acts in the brain and then what is going on in the brain when a human is desiring to drink, we can look in our animal models. So you can look within a laboratory rat, for example, at the exact moment when that animal suddenly makes the decision to go get a drink. Maybe it's a big drinking animal, you know it likes to drink every day. You can look at the exact moment when it decides to walk over, press the lever, and get those drinks of alcohol that it gets to get in the lab. And that lets us discover which parts of the brain work for that exact time when the animal makes that decision, which parts of the brains are activated. We can see it's the same as in the humans. So now we can use these amazing tools from the Brain Initiative where we can record the activity of thousands of neurons at the same time across the brain and determine what brain areas and neurons in those brain areas are involved in the decision to drink. For example, which brain area starts it all off, which is the place where that important trigger happens. And so we are discovering now which part of those circuits are critical. And then this really relates to something we've just heard about with these new technologies to look at the kinds of cells you're recording from in the brain and understanding what behavior they relate to. You can now understand what the genetic profile of those cells might be. That can give us new tools to target just specific subsets of cells in the brain that are responsible for driving the next drink separately from messing up the function of the rest of the brain and the rest of our behaviors. So this is really where our lab and many labs across the country are focused on and trying to understand addiction is how can we very selectively with these very important new tools affect behavior such that we can help people control these urges to drink and not mess with their other day-to-day behavior like some of our old-fashioned drugs.
Yeah, I think specificity and understanding is the key. So why don't you tell the folks what you've bet the rest of your scientific career on?
Well, with this reference that I described at the beginning here, we can take a bit of a different approach to both understanding and treating disease. On the one hand, we really do have a reference that we can map disease data to and understand which types of cells are affected over the course of disease. So this has actually been work that's stimulated by the National Institute on Aging. We run a program called the Seattle Alzheimer's Disease Brain Cell Atlas, or CAD. And the idea there is to now scale these approaches to be able to look at cohorts of individuals spanning the spectrum of disease, to be able to map their disease progression, and to look for cellular effects that happen along the course of disease. And the idea is to look for the earliest effects when something could still be done. And it turns out that this strategy really works. You can get access to these tissues, you can map them, you can then identify very specific types of cells that are lost or otherwise affected in disease. And so, for example, in the case of Alzheimer's, where most of the work has been focused on amyloid plaques and neurofibrillary tangles, other things are going on too. We're losing very specific types of neurons at early stages in disease, and that probably stimulates a cascade of events that eventually leads to cognitive decline. But it starts in a very, very specific way. And this really gives this idea of this cellular vulnerability early in disease. Now that couples with the other big advance coming from the Brain Initiative, which is that because we're defining cells by the genes they use, this means that the genes are tightly regulated to be active only in those cells. We can now identify those regulatory elements, so-called enhancers, these can fit into a virus as a delivery vehicle, and all of a sudden, you have a tool to be able to selectively target a particular cell population and deliver a therapeutic payload in a genetic therapy. So this is precision medicine that you can do. So we, all of a sudden, because of this reference, we have the tools to be able to identify the vulnerable types, and we have the tools to be able to target those vulnerable types. And this opens up a whole new avenue for being able to go after cellular therapies, and because the cells give rise to the circuits, circuit therapies. And so this is, I think, something that we're going to find again and again and again, that we can't think of a pill for everything. We have to think about what part of the system is affected, and we can target that vulnerable anatomy now.
So what diseases are you targeting first? What's on the top? What's at the top of your list?
So our top list, which may not seem like a very small list, but it's the major neurodegenerative diseases. So Alzheimer's and related dementias, Parkinson's disease, Lewy body disorder, ALS, and Huntington's. And the reason for this is that they are all neurodegenerative diseases. We already know that there's some degree of cellular vulnerability in all of them. But when we have these techniques can really throw it at this, we can understand the sequence of events with much greater specificity. So they really align up well with the tools.
Yeah, what's really fascinating, but also devastating about these diseases is that they're very specific cell types of these 86 billion of many thousands of types that are vulnerable. And I think the work you're doing is identifying what makes them differ from each other, and therefore, hopefully, strategies within our lifetime to actually protect them. So switching gears a little bit, Dr. Morell, can you tell us about the work you're doing with NeuroPace, why you left the great academic world to be involved in this amazing enterprise, and the partnership you've had with the Brain Initiative to treat young kids?
Yes, and the Brain Initiative has been so important, and I will get to that. So it was interesting to hear about targets and circuits. It's the same thing for epilepsy. Seizures don't begin all over the brain. At the same time, they begin in a focus. And to some extent, everybody's different. So this personalization is important. And our concept was, since this is a disorder of electricity, that's what causes the seizure, that perhaps the approach would be to fight electricity with stimulation. And as I mentioned, the analogy with the heart. So this project started the same year that Google was incorporated. They were our next door neighbors, so it's been a long effort to develop the technology, which was in part funded by National Science Foundation, and then to develop the clinical application, and that was initially funded by private monies, by the investment community, which fortunately we have a strong ecosystem in the United States and America to do that. We did clinical trials beginning many years ago in the most common type of epilepsy, and we found that by targeting the patient's seizure focus and recording the data all of the time, sensing continuously and recording, that we could identify when seizures were about to begin and provide stimulation to terminate it. And so NeuroPace, the name of the company, developed the RNS system, and this was approved by FDA for treatment of focal epilepsy in 2013. There are now over 8,800 people who are treated, and these individuals are providing data. All of this data, and almost everybody opts in, is stored in a secure data set. And we have been able to look at this data set, and there are now 36,000 patient years of data and 26 million samples of EEG, intracranial electrical activity, as people are walking around living their normal life and being treated successfully. So we got to the point where we recognized that there are other types of epilepsy. They're not as common. And as a little company, we did not have the resources to study these. So the Brain Initiative has an extraordinary program called the Public-Private Partnership. And we were able to achieve or access funding from this to study this less common and horrific epilepsy, which I will talk about later, called Lennox-Gastaut. We used the information we had from the other type of epilepsy to investigate biomarkers and understand networks. And that helped us understand in the Brain Initiative with the Brain Initiative funding, how to place leads for the neurosurgeons and how to program the stimulation intervention with the neurologists, all the while continuing to get data that has allowed us to learn about a different epilepsy that has some similarities but is not exactly the same. This would not have happened without the Brain Initiative. We didn't have the resources. Based on the results of this study in Lennox-Gastaut, there is enough information about the safety and the viability of this, the effectiveness of this treatment, that our little company is able to find funds to conduct the kind of trial that's needed to move towards FDA approval and broader availability.
Yeah, it's really impressive. You know, when I first started the Brain Initiative, I think your grant application had just come in, and you were scrutinized very carefully.
I imagine.
You remember, I think,
Yeah.
for safety, but, you know, you convinced us. And also, then the now former director of NINDS told me, Dr. Walter Korosec, she said, we got nothing for these kids. We got to do something. We got nothing for them, and we'll get back to the success there. We mentioned biomarkers, Dr. Data. When did I start calling you Dr. Data? Tell us about your cool motion sequence AI tools. It's really quite amazing.
Sure.
Tell us a bit about it and how it's being applied.
All right. Is it okay if I tell you how science is actually done a little bit? Because I think it'll be fun, right? So my lab has long been interested in trying to understand how the brain builds body language. But the problem is, like, how do you describe body language to use it as a biomarker? Like, how would you describe my behavior right now? How would I describe yours? It's hard to come up with language, and it's very difficult to do that objectively, right? And so my lab for many years did what everyone does, which is to take videos, right, like with your cell phone, of animals or people behaving in 2D, and then I'd like pay some undergraduate $10 an hour to like write down what they saw. Then one day I was hanging out with a graduate student who was doing this kind of work. And I was asking him what he was going to do over the weekend. And he said he was going to play some video games. And I was like, I was trying to be an interested advisor, and I was asking, talking about what video games he was playing and why he enjoyed them. And it turns out he was playing what was then the state-of-the-art video game console, which was about 10 years ago, which is called the Microsoft Kinect. The Microsoft Kinect is cool because it has a camera that faces outward that images you, the human, in 3 dimensions. And so you kind of can play tennis, or you can play hockey or whatever. And I thought to myself, well, wouldn't it be great if I could pop the sensor out of the Kinect and point it at animals or people to understand their body language, to measure their body language in 3D. So I gave Alex $100. There was a Best Buy down the street. He ran over, he got a Kinect, and we put it over a mouse. And it was amazing watching this first video come off of the Kinect. You could see all of the little gestures the mouse was making as it was exploring an arena. And so this was really transformative to us. It told us that if we could look at body language in 3 dimensions instead of 2, we might really be able to understand the way the brain builds behavior. But then we had another problem, right? Which is that body language is super complicated. How do you objectively characterize it and quantify it? It turns out that the folks that study actual language had just begun to use machine learning, which was pretty new in some senses back then, and artificial intelligence, to discover the parts of language. So we adapted those early machine learning tools for understanding human speech and applied them to these behavior videos, these 3D behavior videos that we got from a video game console, and it worked beautifully. We had a beautiful description of the behavioral syllables and grammar out of which mouse behavior and later human behavior are created and organized. And that has led us down this road where we can give people and animals different drugs and understand how the drugs impact behavior. We can look at mice that have mutations in different diseases. We've studied all sorts of diseases. We studied Parkinson's models, Alzheimer's models. We spent a lot of time looking at autism spectrum disorders, neurodevelopmental disorders. And in all of these different syndromes, in both animals and people, body language differs. And it often differs earlier than even trained clinicians can detect. And so we're really excited about this technology, which basically came because my student likes to play video games, but we brought it into the world, and now it's really an important way of looking at behavior and using it as a biomarker.
So I have two questions for you. Did you charge that console to private money or to one of our grants?
I did not.
Did somebody give you a hard time over it? Okay. Second serious question. I mean, I've been following Bob's work for many, many years. Can you tell us about, what I found fascinating is that he's being modest. You were able to break down complex behaviors into a set of discrete units and to devise this theory that there's a syntax to our behaviors. Can you talk about that? Because I think, for example, in the case of your dad, if we were able to break that down, it's not just the neurologist saying, yeah, I knew something was up, but he might have been able to point at something very specific and a more definitive diagnosis so that one day when we have a protective cure, right? So tell us a bit about the syntax of animal behavior.
Yeah, so both animals and people build their behaviors out of stereotype parts. And so for us, the challenge is how do you identify those parts? Again, things that we call behavioral syllables. And those parts are sequenced together by the brain in time. And part of our Brain Initiative funded work has been to perform this technique, which we call motion sequencing, in combination with brain recordings to better understand how the brain actually controls which syllable you express at any moment in time and how the brain puts them and assembles different sequences to allow people and animals to reach different goals and to express different actions. And all of that, all those neural mechanisms are exactly those that often are affected during disease, which is why this is so exciting, because we think that by looking at syllables and grammar of behavior, we're really kind of taking a sneak peek under the hood at the function of the brain.
So if we have better data for behavior and we can tie it to specific cell types and we know how to intervene, I mean, seems like science fiction is coming to us today. We were at a conference over the weekend, among present company accepted, among the best and brightest minds in neuroscience, and one of the more accomplished and brilliant, don't tell them I said this, computational neuroscientist said to me, because, you know, this is great, but aren't we at the point where if we had all the data, we wouldn't know what to do with it? But I would say that artificial intelligence, this is the good side, because it actually allows us to leverage it. So can you tell us a bit about how you're handling the data that you're getting from all this information from human brains in both health and disease, and how's that going to help us get to where we want to be?
Well, first of all, biology has become a big data field. And there's no way around that. You know, these are extremely high information content data sets. They're extraordinarily large and complex data sets. And machine learning has just become part of the trade now for this and has been for some time. I do think the advent of AI and foundation models is a bit of a different beast now. And we're starting to see that there are really remarkable things that can be done at the level of genes, at the level of cells, at the level or even starting to get to patient-level data that open up new avenues for how to understand the grammar of each of those, but then also coordinate across them. And I think that this is where we're going to need to go now, is to have this multimodal modeling of this sort that starts to understand the whole process. It can integrate across much, much larger data sets. It can start to infer and impute across data sets and harness it for actually useful, clinically actionable things. And I can easily imagine from the kinds of data that we'll be generating that we'll eventually be able to make predictions about things that could have been earlier biomarkers, for example, or mechanisms of disease. The tools of LLMs are already making a big impact in our field. The cell atlases, for example, can now be annotated with the world's literature with LLMs and navigated in ways that, with natural language, that would be impossible for an individual scientist to have done with more traditional means. And now this is starting to get into sort of lab in a loop and people now using these agentic workflows to start to investigate hypotheses and generate the next experiments. So I think we're entering a whole new world here, but it's, the AI is coming into this field, and I think the trick is how to leverage it as quickly as possible.
Yeah, and we've hit an interesting crossroads where the projects you're describing were, and the technical know-how and the output has been the result of team science. And what we learned is when you scale up to a certain point, it just becomes a whole different world. And now AI is going to take us even deeper into that world. Let's talk a little bit about model systems. You know, animal models and models in a dish, as it were. Oftentimes, we've, in the biomedical community, have been accused of curing cancer in a mouse, but not helping a human. And I think that's, to some degree, that's fair, but I think there's a reason why mice, rats, rodents are different than humans. But that actually can tell us quite a bit. And I think it's been critical to use these models in the addiction field. So can you tell us, what are the pitfalls, what are the advantages of using, say, a rodent, not just curing alcohol or tobacco addiction in a rodent, but actually as a pathway toward helping humans? Yeah, sure.
I think what's really critical is having the data at the human level so that when you use this very simple system, like the torpedo electric organ that's loaded with these nicotine receptors, you know what parts you can take all the way to the human brain and what parts you have to just sort of leave behind. That requires all of this molecular information, so sequencing the human genome allowed us to know where the point mutations were between a human, a mouse, a torpedo fish, and therefore where we could target and where we couldn't. So for example, there are plant drugs that target particular nicotine receptors in the mouse. They decrease appetite in the mouse. It was very exciting. Could this same plant actually decrease appetite in a human? There's a point mutation, and we knew from the molecular modeling that that plant substance didn't fit into the human receptor the same way it did with the mouse receptor. So that's something you wouldn't take into the clinic. And on the other hand, the parts of the mouse nicotine receptor that were important for addiction were very conserved, and that molecule could be taken into the clinic. That's one example on a very small data, small science level. Now imagine that you can scale that up because of the tools of the Brain Initiative to every comparison between mouse cells, human cells, mouse molecules, human molecules, and maybe even torpedo electric fish cells and torpedo electric fish molecules. That's where we have the power now to really leverage, where you can change things, do those experiments, not mess up a human, nobody wants to mess up any humans, and then take the promising targets safely into the human system.
The one thing I would add that goes hand in hand with what you said, Marina, is so many of the things that we're interested in looking at for human health involve human behavior. And right now, you can't study a behavior like drug-seeking behavior or something like a memory in cells in a dish. Right now, we need a whole organism that can show us the behavior so we can understand the biological basis for it. So in that sense, the model systems are still really necessary. Someday, we may understand how brains work enough. That's our eventual goal. But right now, we're still trying to understand how these billions of cells work together to produce these important behaviors.
Yeah, I think the comparative approaches are really important. I remember in the early days of the Cell Census Project, back when I was a civilian with the UN, we were thinking about, well, how are we going to keep the bosses happy? Can we give them something actually useful? And it came down to really good biology, and that was, let's compare the cell types between different species of animals. And what we would learn about that. And, I remember when we wrote the paper, at least a light bulb went off in my head, but I think you turned the switch on. You want to talk about that?
Sure. Well, one of the real beauties of studying based on the genes is that we can also align across species quantitatively. And so as we first started to discover that we could define cell types based on their molecular profiles, one of the first things we wanted to do is to compare a mouse and a human. And the results were pretty amazing, because down to a certain level of detail, granularity, you can sort of think of this as a hierarchical organization. Down to a certain level, we're the same as a mouse. Like, it just doesn't look any different. That's not to say that the cells are the same. They can vary in their properties and things, but the same types of cells are there. When you go down to a very fine level of detail, now you start to see species differences. You see expansions of certain kinds of cells, and of course there are other ways that you can use the same building blocks and make things a bit different. But we can align across species, and now we know that we can do this across all mammals. And this is essentially true, that down to that level, there's a mammalian blueprint. Now, on the other hand, things that might be druggable targets can actually be used very differently in those same cells. And so we have to have this information. We need this, and we've had a bit of a knowledge gap in human. But at the end of the day, we still need model systems. We need something where we can go and actually perturb the system, where we can develop things, where we can study behavior. And so this isn't a either or, this is an and situation.
Yeah, let's shift gears a little bit. Well, before we shift, let me just, I hope you all can feel the passion and just the creativity. And the two things together are what's really driving discovery here. You know, one thing we like to say about ourselves in the U.S. is our innovation, our innovative and entrepreneurial spirit. And you know, I'd like to hear a little bit about how this comes about. Dr. Morell, you know, you're with NeuroPace. I remember when NeuroPace was a startup company. I remember when NeuroPace was trying to get that FDA approval. Tell us a bit about that journey, about what it takes to push these ideas forward, having to keep investors happy, having to stay involved, needing to get over the commercial.
Well, and this is really translational research. We were talking about models. There's not an animal model for Lennox-Gastaut system. There's symptoms. Seizures explode. They are drop seizures and convulsions. They are multiple times a day. They destroyed families. The children lose development. But we didn't know what was going on in the brain. We know that there are a number of different brain abnormalities that could cause it, like malformations of cortical development or tuberous sclerosis or diffuse brain injury, but we didn't know what was going on in the brain. And in order to understand how to treat it, we had to spend a lot of money to develop the technology. And as somebody not familiar with business, I thought, wow, we're spending millions of dollars of other people's money to develop something that we have no idea if it's going to work. So it was a big deal, and the investors in NeuroPace early on took a big risk. And we didn't know how hard it was going to be, which is often the case because it would be difficult to think about doing it if you knew how difficult it was. But it's a mission, and everybody in the company has the mission. Then those many, many years, and finally getting FDA indication for use, and then being able, the company, to put money back into research and to continue to fuel, because everything we learn about one patient informs the next patient. We are still small. We're getting a little bigger as we go along, but it has been a very big pull. Also educating and all of educating, and not just the patients and their families, but physicians, to think about treating a disorder in an entirely different way. So this kind of shifting the paradigm is also one of the challenges of companies that are being really innovative. And this is a US-based company. The trial we did that the Brain Initiative funded was a collaboration between NeuroPace and 7 academic centers, 7 clinical centers, so pulling together all of this brain power and all the people contributing all of the hours to understand how to address this absolutely devastating disorder. As John said, no treatment for it.
Yeah, it's really a remarkable journey. And through your Brain Health Accelerator, I know you have your sights on some pretty big goals. And I've heard you say you might think we'd have a clinical trial within 5 years. But you've already spun off a company. You want to tell us about that?
Sure. So the company that John is referring to is called Epicure, and this is a company based on the premise of trying to treat childhood epilepsies where the genetic cause is known. In this particular case, there are multiple childhood epilepsies that fall into that category. Dravet syndrome is one, as a haploinsufficiency of the SCN1A gene. It's another called SLC6A1 disorder, another called SYNGAP. And in each of these cases, we know the gene that's affected, we know the cell populations it's affected in, And so it kind of provides a path to trying to restore or augment functional gene expression back into the red cells. And we have terrific mouse studies that now show that by restoring such gene function, we can ameliorate most of these symptoms, including childhood mortality, including epilepsy phenotypes, but only if we do it in the red cells. If we put it into a more generically, we get off-target effects and actually worsens phenotypes. So I believe that this is going to be more the norm than the exception in the future. But that's one example where we were able to take sort of some of the first-generation tools to target quite broad classes like inhibitory interneurons and use it to try to have a gene replacement approach in these scenarios. And it works well enough, we really feel that this can make it into the clinic soon. So that's on a path now to try to spin out and actually see that happen. I think there will be many other opportunities here, and there are low-hanging fruit already where we know diseases, where we know the most vulnerable cell populations. ALS is a great example, where it's such a tragic and rapidly advancing disease that there's an incredible need, and people are willing to try more experimental therapies, that we can target the motor neurons that are affected and do that very, very selectively. But this is also true for dopaminergic neurons, for Parkinson's. I mean, really, we have the toolkit now to target the vulnerable populations in any disease. And so at the outset here, we're seeing this as more of a partnership model. Let's work with the groups that have targets that they would like to deliver and have patient populations who are willing, and we bring the specificity to this.
And don't forget brain donations, so we can study the disease and identify those targets, yeah?
We
I don't have to tell you that.
We have to have brain donation.
So, I hope we're all appreciating that knowledge is power. And to that end, I'm sure, I don't think anybody has not heard about these amazing GLP-1 drugs in changing people's lives in terms of controlling diabetes, weight loss, but also, as it turns out, and you might be reading about this, the effects of certain GLP-1 drugs on addiction, alcohol use, reward and depression, and the variability that we're seeing amongst those. But now I think we have the tools. So how has this affected your research in the addiction field? And how can this guy help you have a better understanding?
Marie, Dr. Pacheco has an actual ongoing study on this.
Yeah, we're looking at this right now. So anybody here hacking your GLP-1 system? I see you. So one of the things that's been clear is that certain individuals have this remarkable lack of craving. I've had a friend who told me, I've craved foods that are bad for me my whole life, and I started on GLP-1, and I've been able to control them. And it's not just controlled my diet, my diabetes, but it's actually changed how I go about my life. You've probably also heard that there are individuals, many on antidepressant medications, who people say they crash. What does that mean? It means that when you decrease the motivation for all of these things that are perhaps driving you to get up out of bed if you have a history of depression, now they are not motivating you, and in fact, your depression can be much worsened. So there's a real risk here for some individuals to have a very adverse outcome. Your brain atlas is going to tell us exactly which cells express which receptors, not just for GLPs, but for the other targets that are being added into these medications. And what we're trying to do now in a mouse model is to ask about when you give GLP-1s to animals drinking alcohol, we expect that should decrease their choice of alcohol. But we all know that stress increases alcohol intake, but stress also may be exacerbated by these GLP-1s. So understanding which cell types, particularly those sensitive to stressors, versus those sensitive to rewards, express these receptors is going to be very important in understanding who's at real risk of really crashing if they take these new miracle medications, which for some people are really transformative.
Yeah. Great. I want to touch on a few more things before we wrap up and take questions. Let's hear from you folks. Let's think about maybe 10 years ago. What were you dreaming? And we're scientists. We dream about, oh, could I do this really cool experiment to answer this cool question? But oftentimes we think about a question that we just can't possibly answer. So I'd just like to hear from you. You know, what were you thinking about? Oh, geez, I wish I could solve that. There's no way I can do it. That today, what dream experiment or set of knowledge that you could only dream about 10 years ago are you doing right now? And I'm going to start with you, Bob.
So I think a thing that we would really love to be able to do is to understand how all the different parts of the brain together in real time collaborate to let you make decisions and generate actions. And being able to solve that problem, to understand how the brain works holistically, requires that we be able to record activity, the actions of the brain itself, in multiple places all at once, really fast. And the BRAIN Initiative has been incredibly successful at building tools that allow us to record brain activity. It requires that we have tools that allow us to, while the animal is doing whatever it's doing, and while we're recording neural activity, to perturb neural activity, to inject a little activity in one part or another, to take it away from one neuron or from another neuron, to ask causally about what parts of the brain are doing what. And we need to integrate all of that with a really rich description of the animal's behavior. And I think one of the amazing successes of the BRAIN Initiative is that this dream that I had 10 years ago when this whole thing started seems like it's coming to fruition, that collectively all of us now are being able to think about the brain not as one part or one neuron or one system, but really holistically. And that's going to be key if we're going to treat diseases, because most diseases of the brain affect the whole brain.
Patricia?
I mean, it's very similar. So the BRAIN Initiative tools are letting us record from many, many, many thousands of neurons at the same time. And because of that, you can get a snapshot of the brain activity at any moment. And that snapshot means something. So in the context of addiction, it could mean the animal's about to go seek the drug. So because of the ability to measure neurons and then use the computational processes like AI and different kinds of machine learning processes, we can look at what the brain's doing and predict what the animal's going to do in the next few minutes, maybe the next second, the next 60 seconds. So being able to decode brain activity states, like in an Arnold Schwarzenegger, science fiction movie, we're actually here now. And so now being able to use that ability to decode exactly what you or the rat is thinking in a way that can help us treat human disorder is really the amazing goal that we all wanted to get to, that I think we're really at the precipice of.
I'm going to talk about this as an epilepsy doctor. So patients come into the clinic, and I say, how many seizures have you had? And they say, well, I think I've had 3 a month. But sometimes people don't remember their seizures, or sometimes they don't want to keep track of them, or sometimes they're asleep. And so it's a black box. It's a black box making big decisions based on subjective, and we know, despite the patient's best effort, flawed information. So now, with this device, I have learned a lot about epilepsy in general, but more importantly, I can see what's going on in my patients. I can see if how I have programmed stimulation is actually having an effect. I can see whether there's improvement in the brain electrical activity over time. Am I trending in the right direction? I can see many seizures that they don't know that they're having, and abnormal electrical activity that may not rise to perception of a seizure, but is probably explaining why they are tired, why they are depressed, why their thoughts are slowed down, why they're not 100%. And so it makes me a better doctor. And when I see this data on our database, I can swing that computer screen around in the clinic, and the patient can see what is going on with their own data, and it's revelatory to them. It's the first time they've known what's going on, and it's very empowering. So to see this, the tool for for the physician and the patient this information that enables dynamic interactions has made treating people with epilepsy is just not a mystery like it was.
Yeah, that sense of revelation, that you knew something was there, but you couldn't see it, and now all of a sudden, that's got to be quite amazing. Marina, what were you dreaming about 10 years ago?
Well, what they said. I look at what my students can do on a daily basis in terms of figuring out tools that they can just pick up and use to ask any question about which brain area is involved in which behavior. The things that they can ask that are almost immediately relevant to psychiatry particularly, but in general to how we behave, has made me basically get up every morning. There are questions that we just couldn't even have imagined asking, and the tools enable us to be more bold and enable my students to be incredibly creative in ways that they could not 10 years ago.
Ed, you're living the dream, but you're living the dream on behalf of so many people.
Well, for me, I think 10 years ago, I couldn't have imagined that I was talking about treating brain disorders. Like, we were just not there. You know, we were at sort of I was in an esoteric field of mapping brains with different methods that were kind of underpowered at the time.
And some people thought it was boring.
Well, they were just not very
They were
creative.
wrong.
And-
They were wrong.
But, you know, I think, and I really want to give Brain Initiative a lot of credit for this. You know, bringing the new methods into this really started to change the field in many ways. But one of the ways is that we have a way of describing the brain that links across scales now. So describing cells by genes gives you this incredibly important locus where the cell is the basic unit, but you know the genes that give it its properties, and the cells give rise to the functional circuitry. And so it's not solving one problem, it's solving three problems. They become the same problem now, just understanding the cellular and circuit organization of the brain. But the real kicker was being able to manipulate. We just saw this as descriptive, and it's no longer descriptive. It's manipulative, and in the space of medicine, this is now precision medicine. And so I think it's just been profoundly career-changing for me to see that we're kind of at the forefront of having this understanding, but the understanding came with tools, and that's actually actionable immediately. And I think that we just have to pave the way and keep this barreling through, but I think it's going to change the whole field.
Yeah, I think with folks like we have here on the panel, we can literally invent the future and bring it closer to us. I want to leave some time for questions, but before we go to that, Dr. Morell, you know, I've known a few child neurologists, and in particular those who care for patients with epilepsy. And what really struck me over time is that you really do develop a bond with these patients because you see them when they're little, you're really trying to help them develop as normally as possible, and often you follow them into young adulthood. Can you tell us about your experience with that and how it influenced your transition to the neurotech world?
Well, and families too. As a kind of applied scientist, that what drives you is relieving human suffering. That's why we are all here. And so I mentioned that I was at Columbia, and when I decided that I wanted to see if I could make a difference much faster by joining NeuroPace, high risk, but could be big gain, I had to say goodbye to my patients, and that's really hard. Because the patients and their families, you get very close with them. And there was one patient that it was particularly hard because I hadn't helped him. It was a patient with Lennox-Gastaut. I'd been taking care of him for 5 years. I tried everything. I hadn't helped him. So we're still bonded. I told the family and the patient, there's always hope, there's always hope. And then when I went to say goodbye, they said, where are you going? And I said, I'm going to go join some engineers who are going to try to learn about your brain to create a new treatment. And it turns out this patient was the second patient who enrolled 20 years later in the NIH-funded, the Brain Initiative-funded trial. And he's done really well. So I got a letter. We were all surprised, the family and me, that our paths had crossed. I got a letter from the family, and it was very sweet and very funny. It said, "Dear Dr. Morell, we guess we're glad you left."
So, thank you all for your insights and for all that you do. You should know that we all keep a box of tissue by our desks at NIH, because when we hear these stories, we really feel it.
We want to extend our deep appreciation to Senators Schmidt and Blumenthal, along with the Congressional Neuroscience Caucus, for their continued leadership and steadfast support for the BRAIN Initiative. And with that, we heard a lot today. We heard a lot about brain circuits. We heard a lot about a lot of sciencey stuff on biomarkers and diagnosis. But the translation of those discoveries, it was the way it can change human health, it can change the way that we live, and it can change the way that we heal. And so with that support is what we want to keep supporting, the support that comes to the BRAIN Initiative, but even more so of keeping that innovation here in the United States and be able to keep propagating it in our society. And so we respectfully urge Congress to provide the strongest possible funding for the BRAIN Initiative so it can continue to deliver these transformative discoveries and transformative treatments to bring real-world impact to patients, families, and to those within our nation. So thank you very much for attending. We appreciate you taking the time, and we hope you will bring this back to our offices and continue to support this research and the initiatives that we have here in human health. Thank you very much.